Just hit a demonic back day. Now, it's time to do some polysaccharides. So the last videos I made were on monosaccharides and disaccharides. Gonna need to watch that video, otherwise none of this will make sense. Real quick, bro, subscribe to the video, like all that bollocks. Let's get into it, bro. So here we've got glucose, okay? This is our monosaccharide glucose. Pretty sure we went over this before. You do have to memorize this condensed structure, okay? So this is kind of the simple version of the structure that you need to learn. Now, of course, it's our hexose sugar. It has 6 carbons, 1 here. There's 2, 3, 4, 5, and 6. Each little vertice, each corner, contains 1 carbon. And this is something you'll learn in A-level chemistry, in organic chemistry. But for now, if you're not doing that, just learn that each corner has 1 carbon. Now, you'll also remember it does also have our hydroxyl groups, which are the ones involved in our chemical reactions. It's also the one that is responsible for its reaction, its solubility in water. So that's something you need to remember. And now the point of me getting you up to speed with all this is because this is glucose, yes, but it actually has a gangster name, right? It's street name. And we call it alpha glucose. Reason being is this carbon over here, carbon 1, you see the hydrogen is actually above the plane of the ring. It's above the carbon 1. It's on top, so we call it alpha glucose. That's its gangster name. And now here we have its weird cousin, beta glucose. And now the thing with this is the hydrogen is on the bottom. Now this is actually very important because they're going to have very different ways of reacting. And it's especially important in polysaccharides, and we'll get to that in a second, but understand this, bro. So they actually have the same molecular formula, right? So they have 6 carbons, they have 12 hydrogens, and 6 oxygens. It doesn't actually matter where the hydrogen is positioned, right? They still both have the same molecular formula. And the thing is, if I was to hide them, you wouldn't be able to tell me which one's which, right? And the thing is, we call this an isomer. They're essentially different versions of each other with a slightly differently arranged structure, right? How do I analogize this? Let's say I had 2 houses, right? And they were both made out of 10,000 bricks, right? And one of them had 2 bedrooms and the other one had 1 bedroom and a hall. They both have the same amount of bricks, but they both look differently in a sense. That was probably a horrible analogy, but essentially what you should understand is that they both have the same molecular formula, they both have the same amount of bricks, but one of them looks differently. And because of that little difference, they react differently. And we call that an isomer. And now you actually have to learn this definition, bro, otherwise you are completely cooked. Now the definition is this: it is a molecule with the same molecular formula, but a different structural formula. So that's basically what I was just saying, right? It's not too difficult to get your head around, but the difficulty comes more in memorizing which one is which, being able to spot the difference, kind of like Where's Wally, right? You need to tell me which one is alpha glucose and which one is beta glucose. So remember, I did say alpha is where the hydrogen is on the top, so therefore it's the right one. And beta is the one where the hydrogen is on the bottom, so the left one. Now, they're going to be asking you this a lot in the exam, but where it mostly comes into play is polysaccharides, because like I said, these react differently, and we'll get to that in just a minute. So a polysaccharide, right? It's a massive carbohydrate consisting of many repeating units of the same thing. So let's say we have 2 molecules of alpha glucose and we're reacting together. We react them together and we form a glycosidic bond. Now they still have available hydroxyls, right? They can go again, right? They don't just stop at disaccharides, they can continue going. So of course more monosaccharides line up and they have their hydroxyls and they form their condensation reaction. They release their water and they get even bigger. Now a bit like your mom, it's massive now. It can continue getting bigger and bigger and bigger. It forms this massive chain of monosaccharides repeating over and over and over again, which we call a polysaccharide, right? Poly meaning many, saccharide meaning sugar. And again, it's just a very easy definition, learn it. And this is one of the things we call a polymer, right? A polymer is basically just a massive molecule consisting of the same things repeating over and over again, right? A polysaccharide is a polymer. DNA is a polymer. Protein is a polymer. There are so many different things in biology that are a polymer. And a carbohydrate just happens to be one of them because it's a massive molecule consisting of the same things over and over and over again, which is your sugars, right? Your monosaccharides. Gotta learn the definition, little bro. And it's it's pretty easy. Like we've discussed, just a large molecule consisting of smaller repeating units called monomers. Now the thing is with alpha and beta glucose is that they can go on to form different types of polymers. Now when we're talking about alpha glucose, they can go on to form starch. Now starch is a polysaccharide and it's used by it's used by plants. Now the thing is with polysaccharides is when a particular organism needs energy, it's better if we store the sugars as a massive chain, and then when we need it, we can just break it down and use it as energy. And starch, in this case, happens to be the energy storage for a plant. Now, the polysaccharide starch happens to exist in two different kind of molecules, two different versions, amylose and amylopectin. So let's talk about amylose first. So amylose is a polysaccharide made out of alpha glucose. I did kind of just show you this, but this is kind of what amylose looks like, or amylose. It's not Amy, it's you know what, just whatever. Amylose is a straight chain alpha glucose polymer. In this straight chain structure, what you'll find if you zoom in is they start to form these little hydrogen bonds in between, and this causes it to actually coil. And believe it or not, bro, it's actually really good for it to coil because it makes it more compact, and that's a very good storage molecule. It can be stored really well. The plants can use it very easily to get out their energy, and the most important thing is that it's actually insoluble. It doesn't dissolve in water, it's insoluble, and that means it doesn't affect the water potential, which is an unknown term, but we'll get to that in a second. But it's insoluble, it doesn't dissolve in water, and the reason being is because when you form this massive polysaccharide, you're reacting all these hydroxyl groups together. And as you're doing that, as more and more hydroxyl groups are getting used up, right, there's so few of them that they can't really form these hydrogen bonds with water anymore. So many of them are used up. So many of them are, busy, and they can't form these hydrogen bonds with water anymore. Therefore, they are used up. Unlike your monosaccharides, unlike your disaccharides, these guys, they can't dissolve in water. So remember that, bro. Polysaccharides are insoluble. Now let's talk about amylopectin. So amylopectin is also another polysaccharide made out of alpha glucose. And this here is the polysaccharide, and it's obviously in its condensed form, right? I'm not showing you the other elements, but for this example, I'm going to show you some of the other elements. So on this sixth carbon, there is actually another hydroxyl group. And the thing is, another chain, another sugar can come along and react with it from the top. And so it forms its glycosidic bond, but this time it's a little bit differently positioned. So before, we had a 1-to-4 glycosidic bond where they reacted more level with each other, like adjacently, and that was a 1-to-4 glycosidic bond. But this one's a bit different because it reacts from the top, and we call this a 1-to-6 glycosidic bond. So if you can look at it closely, you can see the top monosaccharide, the first carbon, its hydroxyl group is reacting with the sixth carbon of the saccharide on the bottom. So that's why we call it a 1-to-6 glycosidic bond. It's just trying to illustrate the position of what's reacting. So this reaction could continue going, and you'll notice it's branched off. And that's the special thing about amylopectin. It branches, and you gotta learn that, bro. It branches out. And the reason why it makes it such a good storage molecule for energy is that not only is it insoluble, just like the last, but it's easy to break down. See, the thing is about enzymes is they usually act on the ends. So enzymes are things that basically catalyze reactions. They break things down. And enzymes, which you'll learn in a different topic, but enzymes, if you can remember from GCSE, is they break things down and they act on the ends of things. So the more ends there are, the quicker it can break down. So as you can see in this image, there's so many different enzymes on all these ends, all these different branches, and it makes breaking down amylopectin so much easier. So when the plant needs energy, it just sends all these little, these little goons, these little enzymes. And because there are so many branches, so many ends, these enzymes have so much area to act on, so much surface area to act on. So it breaks it down so much quicker, the energy is released so much faster, and therefore your plant gets the energy so much easier. Alright, that's part 1 then. I'll see you for part 2, bro.